Hello there. I'll start by acknowledging my collaborators on this research, David Gurnant, Paula Marquardt, Brett Tulifsky, Kevin Potter, and my Hendrix College undergraduate researchers. So I'll be highlighting examples of how recent research from my lab and others can help frame new questions. One, especially involving Jeffrey pine, another, the trees lumped together as the ponderosa pines in the US. So these phylogenies of 130 individuals represent 21 taxa, and there's 700 nuclear genes on the left and nearly complete plastid sequences on the right. You won't obviously be able to see details, but the nuclear and plastid tend to be congruent. I just want to tell you that clade resolution informs how we should be recognizing some taxa. First of all, Jeffrey pine is resolved here with the other red taxa in the Sabiniani, which are the California big cone pines. I'll come back to Pinus jeffrey in a moment. The other is that the ponderosa pines west, in the strict sense, are not sister to the ones east. So Pinus brachyptera and Pinus scapularum were originally published separately, but they've been traditionally lumped in the USA as varieties of ponderosa pine, and they are not. The other thing I'll tell you is that we had limited within population sampling, but where we did, for example, where those red stars are two individuals from an isolated stand in the Spring Mountains of southern Nevada, and they resolve in different clades. So this species tree makes it easier to see the resolution of Jeffrey pine is with the coulter pine, gray pine, and Torrey pine clearly. Also, the separation within what is traditionally ponderosa pine goes all the way back to this node B, so the scapularum and brachyptera resolve with all of these mostly Mexican taxa. So most of the nuclear and plastid are congruent, but there's a few examples like Pinus arizonica where they differ. So this is where the pines east of the USA Great Basin are resolved in both nuclear and plastid. So brachyptera is in blue here. Those clades are found all the way up into Wyoming and all the way down into central Mexico. And most of those brachyptera are in one plastome clade. So let me talk about scapularum first. It's in mostly to the east of there, and it's not a north-south boundary for the two needle pines. The mitochondrial data that I just popped up on the right is from a different paper, slightly different sampling. Here, the brachyptera is in squares and scapularum a plus sign, and then the colors are mitochondrial haplotypes. So brachyptera mostly has the H3 haplotype, which is in yellow, and that's the ancestral haplotype that's widely distributed taxonomically and geographically, and it's central in the network. The scapularum plus signs have the divergent H6, and that's also been reported up here in some samples that we don't know whether they should be assigned to scapularum or brachyptera. The other thing I'll tell you is that the extent of mixing is unknown. This is an example in New Mexico where a population you would assign to brachyptera based on morphology and on nuclear and plastid resolution is carrying the H6 haplotype. So mitochondrial mixing in Jeffrey pine is amazing. So the J samples here are all 28 Jeffrey samples. The colors are the mitochondrial haplotypes. Excuse me. The H1 is in purple, the H5 is in green. So notice that the Jeffrey pine samples all are carrying a mitochondrial haplotype of either the nearby H1s or H5s. So I want to tell you how clearly gentle Jeffrey is a California big cone pine. So in addition to the nuclear and plastid data, literature search shows 4 synapomorphies that hold those 4 taxa together. So first of all of them have cones greater than 13 centimeters long. So Jeffrey pine's cones are not heavy, and they don't have the outcurved prickles. They have different seed predators, so you would think that would be a good hypothesis for why that's so, but they all have big cones. The seed wing length to seed length ratio is all less than 2, so the heavier or that shorter seed seed wing in Jeffrey pine of 1.4 means that Jeffrey pine is completely reliant on rodents or Clark's nutcrackers for dispersal. That's some interesting new work from van der Waal and other people's labs recently. In ponderosa pine, wind can still disperse those seeds very effectively, and rodents and birds are important for occasional long-distance dispersals. Also, Jeffrey pine and all the others have a heavier seed, so greater than 0.14 grams, where that heavier seed probably helps attract animals as well as reduce effective wind pollination and dispersal. Wind dispersal. And of course, Jeffrey pine has heptane as a component in the monoterpene. So that makes the Jeffrey samples carrying mitochondrial haplotypes a very interesting question. So geographically, I won't have time to show you evidence. I'll just lay out a few other questions for you. So first of all, the Pinus arizonica and Pinus engelmannii were defined in the USA, in southeast Arizona. However, those names have been assigned to some populations down in Mexico, and neither of those taxa are cohesive. And we also think that there's at least one new taxa in southeast Arizona or an outlying population of one of the Mexican taxa. So the continuous ponderosa pine forest here in Northern California, where the Klamath Cascades and Sierra Nevada ranges meet, have some mixing of the coastal Benthamiana mitochondrial haplotypes and the inland ponderosa haplotypes. There's no current sympatry, but there are two areas that are of interest. So in Southern California, there are mitochondrial haplotypes clear out into the transverse ranges that are shared with the pines of the Mojave Desert that you would assign to Brachyptera. The interesting part of that story is that those haplotypes are different than the rest of Brachyptera, and they are actually more closely related to the Scopularum haplotypes only found out here in the Northeast, and to some haplotypes disjunct down in Mexico. There was a region of contact zone in Montana described by Mitten and Latta some years ago. We also found a contact zone in Idaho, and in Idaho, interestingly enough, there are nuclear and mitochondrial sharing, but the plastid haplotypes are coherent with other nearby populations. And then, of course, there's Pinus jeffreyi. So I just want to point out in conclusion that scientific names matter, and that, for example, ecological niche studies of clades that are actually sharing an ancestry would be useful. Political borders don't matter, and we need studies that are not limited to just north or south of the USA-Mexico border. And how organelle integration happens and the retention of those lineages is ecologically interesting. I'll give you the example of Goodbau's work in lodgepole pine. And obviously, organelles are not definitive for these taxa. And that I hope people will design experiments expecting stands of trees that might have mixed ancestry. So that makes it important to use molecular and morphological measurements on the same plant sample, because some of the broad variation that's reported will be clarified when we do that. So thank you very much.